Dear students, in this presentation, we are going to see the gastritis caused by the bacterium Helicobacter pylori. But before I proceed with this, let me tell you a small story in relation to this. See, till 1980s, doctors used to think that if a patient comes with ulcer, I hope you know what is ulcer. If a patient comes with ulcer, they used to think that the ulcer is caused by either stress or diet or some physiological condition that led to this particular problem. So they used to prescribe medication based on that. But two Australian gastroenterologists who were working on this particular problem, that is peptic ulcer, suspected that some ulcers can be caused by a bacterium, and they named it later as Helicobacter pylori. In fact, they could isolate this bacterium from almost 100 patients in their clinic, and they put forward the concept that gastritis can be caused by a bacterium also. This was not readily accepted by the scientists and also by the doctors community. They said it is not at all possible for a bacterium to survive in such an adverse condition because gastritis happens in the stomach. So if a bacterium causes gastritis, then it must survive in the stomach acidic pH. Almost it is pH 2, we know that. So no bacterium can survive such a condition. So they said that it is not possible. So what Warren and Marshall, the two gastroenterologists, as I said, did was to prove their concept, they made themselves as guinea pigs. Marshall especially took a suspension of Helicobacter pylori in a glass and then he drank it. If his concept was correct, he should be developing gastritis. In just 14 days, he started showing symptoms of gastritis. But of course, because of his immunity, he recovered from the disease very quickly. So, with this particular experiment where the scientists themselves became guinea pigs, they could prove the concept that a bacterium can also cause gastritis, which in turn might lead to peptic ulcer. So after almost 10 years, that is, they put forward the concept in 1982, whereas the concept was accepted by the scientific community in 1992. So after almost 10 years, this concept that Helicobacter pylori can cause gastritis was accepted and treatment was also proceeded in that direction. So in recognition to their discovery, Marshall and Warren were awarded Nobel Prize in the year 2005 in physiology or medicine, and the Nobel Assembly announced that thanks to the pioneering discovery by Marshall and Warren, peptic ulcer disease is no longer a chronic, frequently disabling condition, but a disease that can be cured by a short regimen of antibiotics and acid secretion inhibitors. Because before their discovery, doctors never thought of prescribing antibiotics for peptic ulcer disease. But after this particular discovery, antibiotics started being prescribed as a medication. So with this small introduction, let me proceed further with H. pylori gastritis. As I said, this particular gastritis is caused by the bacterium Helicobacter pylori. Helicobacter pylori is a gram-negative spiral rod. As you can see here, very few spirals are there, but still it is a spiral rod shaped bacteria, and it is gram-negative in nature. The size and the dimension is given here. It is just 0.5 to 0.9 nanometer, micrometer wide and 2 to 4 micrometer long. And this particular feature you need to remember very carefully, that is, it is extremely motile, and the motility is facilitated with lophotrichous flagella. That means a tuft of flagella in one pole of this bacterium. So that is what we call lophotrichous flagella. So this contains sheathed flagella, which makes it very efficient motile organism. And this bacterium is non-spore forming and also non-capsulated. Fine. Coming to the cultural characteristics of this organism, it grows at the temperature of 37 degrees Celsius, and the pH is optimum pH for its growth is 6 to 7. Though we say that it survives in stomach pH, still the optimum pH for its growth is only 6 to 7. And this character is unique to this organism. It is microaerophilic in nature, and it needs around 5 to 20% carbon dioxide and only 5% oxygen for better growth. So it is a microaerophilic organism. It needs very less tension of oxygen in its environment. And this again is a very important character we need to remember. It takes more time to grow and form colonies usually. From 2 to, usually if you cultivate an organism in the laboratory, within a day we get to see the colonies, right? Whereas this organism takes in fact nearly 1 week sometimes, that is 7 days, to give rise to obvious colonies on the plate. And the specific media for its growth is either chocolate agar or Campylobacter media. Chocolate agar is actually blood agar, and this blood agar contains either human blood or sheep blood as one of the component, and it should be heated so that the contents in the red blood cells will come out. So that is chocolate agar. Whereas Campylobacter media, why is it called so? Campylobacter is a genus related to Helicobacter, very much close relative of Helicobacter, and media have been developed to isolate Campylobacter colonies. So the same media can be used for Helicobacter also. And this Campylobacter media basically is a selective blood agar media. What is meant by that? See, blood agar, we know what it contains. In the same blood agar, if you add certain antibiotics, antibacterial antibiotics to inhibit the growth of other organisms, we call it as selective blood agar. So such a selective blood agar is Campylobacter media. And using either of these media, when you grow this organism, it forms discrete domed colonies on the plates. Fine. The next is biochemical characters. Not many biochemical characters we usually use for its detection. Mainly, we should remember these characters. One is the organism produces enzymes like oxidase, catalase, and also phosphatase. And hydrogen sulfide production is also seen. And this particular character again you need to remember. It forms another enzyme called urease, and this urease enzyme is very much useful for this organism. It forms a large amount of urease enzyme. One might wonder why it forms so much urease, but you will understand when we see the pathogenesis of this organism. And another very common organism which is identified using this particular character of urease is Proteus vulgaris. But compared to Proteus vulgaris, this organism produces urease 100 times more. So that much amount of high urease activity is observed with this organism. Okay, let's see the pathogenicity or the disease part. The organism, the bacterium, enters through fecal oral route or oral route. That is, if you come in contact with the saliva of the person who has already been infected or any of his gastric contents, then you might get the disease, or sometimes from fecal route also. Fecal oral route also you can get the organism in your body. And once the organism enters, it is going to colonize and infect the stomach mucosa. You remember this. They don't colonize anywhere else. They directly go to the stomach lining, and in the stomach mucosa they are going to colonize, and the main infection happens there. Fine. Now, what are the various factors that is present in this organism which makes it virulent, which gives it capacity to cause infection in our body? These four virulence factors we need to remember. First is efficient motility by flagella. As I said, this organism is very efficient in motility, and this motility allows the organism to swim to the less acid pH locale beneath the gastric mucus. See, in the stomach, not all the areas of the stomach have the same acidic pH. There are certain areas where the pH will be little lesser. The pH will be little neutral. So, organisms with their motility, with their flagella, can easily move faster and go to the areas where acidic pH is lesser, and this is usually seen beneath the gastric mucus. You will understand it better when I show you the next slide. Just remember it now. The next virulence factor is once it goes to the stomach lining, adhesion to the surface epithelial cells of the stomach with multiple outer membrane proteins. Being a gram-negative organism, the cell wall will contain outer membrane proteins, and with these proteins they can adhere to the epithelial cells lining the stomach mucosa. The next factor is, as I said, it produces an enzyme called urease in abundance. And this urease, what is the function of this urease? You must understand this particular character in H. pylori, which is the principal character. Urease is an enzyme which releases ammonia from urea. Now, this ammonia will become ammonium compound in the stomach area, and wherever this ammonium compound is there, it will neutralize the acidic pH. And this is how, in its immediate vicinity, the organism neutralizes the pH and survives in the stomach. So urease releases ammonia from urea to facilitate survival of the cells in a highly acidic environment. The fourth virulence factor is, after it colonizes the stomach lining, it will release this particular toxin called vacuolizing cytotoxin, VAC-A toxin. This is an exotoxin. VAC-A toxin destroys epithelial cells that it enters, causing multiple large cytoplasmic vacuoles. Wherever it enters, this exotoxin, as soon as it enters, it will cause vacuoles inside the epithelial cells, and it will destroy the epithelial cells. Okay, look at this diagram carefully. The organism enters through the oral route and it will reach the stomach, and in stomach there are certain regions where the bacteria normally colonizes, regions like this, which is shown in yellow circles. And if you just observe one particular area here, this is how it is going to be. So you have the mucosal lining, we call it as mucosal epithelial cells, and on the surface of this mucosal lining you find mucus, we call gastric mucus. This is another special area. This is a highly viscous layer, gastric mucus, and then under this mucosal lining, that is epithelial cell lining, you will see other tissues, right? Usually organisms cannot reach this place that easily, but as soon as it enters, this is the lumen where the organism will first come and get uh, lodged. Okay, once it comes to this stomach region, to avoid the stomach acidity, they immediately go to the mucus region. They don't bind on the mucosal lining. usually they will reside in the mucous region itself. This is a highly viscous region, as I said. So acidity will usually be not there in this particular region that severely. This is one way it avoids acidity. Second is, when it is present in this region, that is in the mucous region, it releases the enzyme urease. And as I said, the urease enzyme will split urea and convert it into ammonia and carbon dioxide. Carbon dioxide gas will be released out, and ammonia usually will neutralize the acidic pH present in this region. So again, this is another way it survives the acidic pH in the stomach. Not only that, the ammonia, when it is released in abundance, will destroy this mucosal lining. This is another way how it causes tissue damage in the stomach. Right? So ammonia is going to be toxic to the epithelial cell lining present here. That is why this epithelial cell lining will get damaged, leading to gastritis and also ulcer formation. And it is one way. The other way is, when it is growing in this mucous region, it also releases toxins, as I said, like vaccae toxin, and this vaccae toxin will enter inside this epithelial cell, and once it enters inside, it forms very large vacuoles in the cytoplasm, and ultimately the epithelial cells will die in this particular region. So this is how it causes tissue damage in the stomach and also survives in the stomach pH. Oh, and the colonization of Helicobacter pylori in the stomach lining results in the infiltration of many immune cells in that area. See, when the organism is invading the tissue, our immune system is not going to keep quiet. It is going to attack this bacterial cells in that particular lining. But while doing so, it also causes further tissue damage of our own cells. Many neutrophils will come, lymphocytes will come, and all of them are going to release different types of cytokines, which in turn not only attack the bacterial cells, it will also cause some amount of damage to our own cells. And it is because of all these effects, that is colonization of the bacterium, act of different toxins, act of urease, and act of our own immune cells, all this will result in different type of effects. So the two main type of effects that you will see in H. pylori infection, one is gastritis. This is the most common effect, where inflammation in the stomach lining will be observed. There will be acute pain in the upper abdomen with nausea and vomiting will be the main symptom that you will see in acute gastritis. But if this gastritis is not treated on time, and if it persists for a longer period of time, then it will result in peptic ulcer, or which can also be called as duodenal ulcer or gastric ulcer. So here there will be ulceration of stomach or duodenum. So these two will be the main effects that you will observe in H. pylori infection. But the more serious effect is seen here. That is, after this H. pylori infection, it could, after a few years, it could lead to these conditions which are more severe in nature. One is, there will be mild chronic gastritis type B that may persist for years or even decades, and most of the times it might be asymptomatic. That is, after the H. pylori infection, even after treatment, sometimes the organism might survive, and it might live in the stomach lining for years together or even 10 years together, and this will lead to very mild disease, or even sometimes it will be asymptomatic, but there will be the infection, there will be the organism, which can be called as chronic gastritis. This is one effect. The second effect is duodenal ulceration, sometimes gastric ulceration as well. That is, both stomach region and also the duodenal region will be severely affected. As I said, it can also be called as peptic ulcer disease. Third very serious effect is this H. pylori can cause chronic atrophic gastritis, from which you will see cancer formation, that is gastric adenocarcinoma, might result sometimes because of this chronic infection. And this is mainly caused by a specific strain of this organism, that is CAG plus strain. So simply speaking, H. pylori infection might result in cancer formation in the gastric region. In fact, this is the first bacterium to be declared as class 1 carcinogen by the World Health Organization. Remember this point. Class 1 carcinogen in the sense the chemicals or any agent that causes that has been shown clear evidence causing cancers in human beings. So this is almost they have identified that 75% of the gastric carcinoma is caused by Helicobacter pylori. So such an effect can be seen in a few people. And sometimes it can also cause B cell lymphomas, again as another type of cancer where B cells will become cancerous in nature, which can also be called as maltomas. That is, in the stomach area we have mucosa associated lymphoid tissue, and this you will study in detail in your immunology part. So even this tissue will become cancerous in nature, so that can be called maltomas. So these are some of the serious effects you will see in H. pylori infection. Symptoms that is commonly observed in gastritis and also in peptic ulcer diseases are like this. The most common symptom is there will be a burning sensation in the abdomen throughout the time of infection, or sometimes even it will result in pain, aching. And second is nausea. You feel like vomiting throughout the day, and sometimes it will even result in vomiting. And bloating in the sense you feel that your stomach is filled with gas. So that will result even in belching. That is, you will keep on um, burp throughout the day. So that is belching. And most of the people will have bad breath when they have this infection. This is mainly because of the release of carbon dioxide and ammonia, the result which is a result of urease activity. And in some serious cases you will also see vomit or stools with blood streaks in it, so bloody vomit or stools. Coming to the epidemiology, H. pylori is exclusively a human infection. That is, it causes disease. This particular species, there are many species under Helicobacter genus, where this particular species, H. pylori, causes gastritis exclusively in human beings. And the global prevalence rate of this infection is almost 30 to 60%. They say almost 50% of the world population has been colonizing this organism in their stomach. And more, the infection is seen more in the developing countries than in the developed countries. This is mainly because of overpopulation and also less hygienic conditions in the developing countries. And the sole source of infection is human gastric mucosa. That is, the source of infection is only from another human being who has already been infected. And the mode of spread is, as I said before, it could be either fecal contamination of food and water or fomites, that is fecal oral route, or it could be direct oral route through kissing or through the saliva contact. You can get the infection. And as I said, these are some important predisposing conditions which will lead to spread of this organism. Poverty, overcrowding, or poor hygienic conditions will favor the transmission. Okay, coming to the diagnosis of this disease in the patient, we have two types of tests. The first one is non-invasive tests. The other one is invasive tests. What is meant by this? Non-invasive means the patient's samples will be taken in a non-invasive manner without doing any biopsy. that means without putting any scopes inside the body. You just take samples from external sources and then see it under use it for diagnosis. That is non-invasive. Invasive means mainly when biopsy is done, endoscopy and biopsy is done, when some tissue from the human body is taken and then used for detection. That is non-invasive tests, invasive tests. So coming to the non-invasive tests, we have 4 important tests here. One is serology, where we are going to detect the antibodies to Helicobacter pylori or its products. For this, you should take blood from the patient and separate the serum and look for the antibodies to H. pylori. It is not very commonly used. It is less sensitive, hence less useful. Whereas the most important non-invasive test that is used in the laboratory is urea breath test. This is the most important test. Here we mainly detect the bacterial urease activity. As I said, this bacterium produces abundance of urease enzyme. So using this particular character, we are going to detect this organism in the person's body. So what this test mainly does is it measures the output of carbon dioxide resulting from the splitting of urea into carbon dioxide and ammonia. This is the urease activity. It is going to break urea into carbon dioxide and ammonia. Carbon dioxide is going to get escaped in your breath. So when they test the amount of carbon dioxide that is coming out in your breath. But how do they do it? Because carbon dioxide is normally breathed out by human beings. So how are they going to detect whether it is a carbon dioxide because of urease activity or because of normal respiration? So for that, what they do is, in the procedure, they first give a capsule of urea which is radio labeled with C14. So it is not a normal carbon. It is a radio labeled carbon that is present in this urea. So in the capsule, such a radioactive urea will be there, which will be taken by the patient orally. So after some period of time, they will detect, or they will measure the isotope of carbon dioxide that is being breathed out by the patient. The device will detect only the radioisotope of carbon dioxide. So with that, if the patient is not releasing radioisotope of carbon dioxide, then there is no urease activity. So then he is not infected with the H. pylori. But when they measure and they detect the radioisotope of carbon dioxide that is coming out, then it is sure that there is urease activity, which is mainly because of H. pylori infection. So this particular test is very sensitive and also very specific, hence it is used commonly in the laboratory. Another test is fecal antigen test. Here they take the fecal sample of the patient and they detect the presence of H. pylori antigens. It could be exotoxins or it could be urease enzyme. So any of the antigens will be detected in the fecal material. This is another option. And the last one is molecular detection, where polymerase chain reaction will be used. So here this is of course expensive technique where they will detect the H. pylori genes using DNA probes. There are specific areas in the H. pylori chromosome. Those genes have been detected and probes have been made ready-made available in the kits, and using these DNA probes you can take the sample from the patient. It could be fecal sample or it could be some tissue sample. You can take any sample. Even stomach washings can be taken and PCR can be run. With this you can detect whether specific H. pylori genes are there in these samples, and with that you can detect the disease. So the invasive tests mainly depends on endoscopic examination. Gastroscopy anta ಮಾಡ್ತಾರೆ. You might have seen in the hospitals where they will insert an endoscope inside your stomach through the mouth. So mouth through ಒಂದು pipe ಅನ್ನು ಒಳಗಡೆ ಹಾಕ್ತಾರೆ. ಅದರಲ್ಲಿ endoscope ಇರುತ್ತೆ. So it will reach the stomach region, and from there they will collect the mucosal specimen, ಅಲ್ಲಿಂದ ಸ್ವಲ್ಪ mucosal specimen collect ಮಾಡ್ತಾರೆ. This is called mucosal biopsy specimen, and using that specimen they can do different things. One is histopathology. They directly go for staining this tissue using tissue stains, hematoxylin ಇದೆಲ್ಲ use ಮಾಡಿ tissue staining ಮಾಡಿ through the microscope they will observe the organism, or they will take the specimen and do simple gram staining and see the presence of gram negative spiral rods. You can even do hanging drop technique for observing the motility. So this is another option. Third is they take that specimen and process it a little and culture it in the specific media that I mentioned, and leave it for 2 to 7 days to observe the colonies. And further, this culturing is mainly done to see its antimicrobial resistance, drug resistance character study ಮಾಡಕ್ಕೆ ಇದು ತುಂಬಾ important. Why? Because in recent years H. pylori also has started showing drug resistance, mainly to two important drugs that is commonly used. So that also must be studied when detection is done. So this is one invasive test. The second also uses endoscopic examination only, where the biopsy specimen will be taken and directly urease test can be done. Whatever I told you about urea breath test, the same technique can be used here also, where direct detection of urease can be done using the biopsy specimen. Coming to the treatment, initially the most important treatment was administration of bismuth salts like Pepto-Bismol, along with 2 antibacterial antibiotics. Why bismuth salts? The reason is bismuth salts believed to act by coating the stomach, and it also has antimicrobial activity. Whereas the antibiotics that is commonly used here for treatment includes metronidazole, that is an azole drug, tetracycline, clarithromycin, and also amoxicillin. And remember one thing, the treatment has to be continued for at least 2 weeks. This is very important. 2 weeks or 14 days treatment must be given to completely control the infection. And nowadays, the patients with ulcers or gastritis, that is the symptoms, a triple combination therapy is used. Remember this particular word again, triple combination therapy. What is present in this is 3 different types of drugs. One is a proton pump inhibitor, that is omeprazole. This proton pump inhibitor is an acid lowering agent. Reduction of acidity increases antimicrobial action, and also the patient will feel better, and so he will be ready to take the treatment, because continuous acidity should be controlled first in this particular problem in gastritis. So for controlling that acidity, this proton pump, what we call antacid, this proton pump inhibitor should be given. One example is omeprazole, and along with that 2 antibiotics must be included. As I said, one is metronidazole and the other one is clarithromycin. And 7 days treatment is enough, which treats almost 90% of the cases. And remember this point, nowadays high rates of resistance to 2 most important drugs. One is to metronidazole and also another one to clarithromycin has been observed in patients. Prevention of infection is only by general measures. We don't have any special vaccines for preventing the infection. So general measures include practicing good hand hygiene, or good hygienic condition is basically necessary. Along with that, personal hygiene, especially washing your hands whenever you use toilets or whenever you are going to do some work in the kitchen. So good hand hygiene is important. And second is unnecessary sharing of utensils or glasses used by one person should be avoided completely. So that's all about H. pylori gastritis.